Okay, so opposite to what Oscar has shown, this is much more like a photograph of a very specific region of interest for us. But just start with a quick background on the species we are working with. The European Seabass is the most important farmed fish in the Mediterranean, over 200,000 metric tons production per year. The biggest player is Turkey, and then we have Greece, Spain, and then somewhere here, Italy. So it's certainly a very highly appreciated fish for human consumption, highly priced, and there has been a lot of work about genetic selection, which is actually what prompted us to work on this species. Okay. And we focus on the most relevant disease. It's an infectious disease caused by a virus, a very small RNA virus, which actually affects also many other fish species. Has a very high mortality rate, especially in juvenile or young adult fish, and essentially targeting the brain, the spinal cord, and the eye. And fish have a very strange behavior, and then they die in high numbers quite quickly in a few days, in a week or 10 days at most. The good news is, at least for geneticists, is that the resistance is to this very severe disease is heritable. It's moderate to, let's say, good. 35% to 40% of the resistance is actually has a genetic component, so there is room for improving fish, making them more resistant to VNN through genetics. And this is what we did in AQUA-FAANG. It's really like a model case for what we hope all the results of AQUA-FAANG will sort of will be used for. So we made an experimental F1 population, crossing in a balanced way 25 males, 25 females of sea bass, and then we did a classical challenge in experimental infection, and we monitor the sorry, I will change this. Okay, I think this is much better. Okay, we monitor. You see, after 5 days, you have a peak of mortality, then you have a slow down, and then it goes to almost nothing. And we monitor for almost a month of infection. So we use this date to death, or just a binary trait, death or surviving, as a trait to associate with the genetic data, with genetic markers. So what we did was to genotype all the fish from this experiment, the parents and the F1 and the progeny, with the Seabass specific 30K SNP array, and then the genome of all parents plus a random, not really random, but representative of most the families of the F1 were sequenced. I think it was 10x coverage, or maybe 15x coverage, if I remember well. So having reconstructed completely the whole genome with their variants, and then the whole genome sequence variants included were imputed using the SNP array data for all the remaining fish. And then using this whole genome sequence genotype data, we did a genome-wide association study, mapping essentially, or actually looking for markers for single nucleotide variants that were significantly associated with resistance, with survival, days of survival to the infection. And you see here, well, the names of the chromosomes are still to be decided, but this is what we call linkage group 12, but it's probably now it's called chromosome 3. This is a Manhattan plot which shows you clearly that in this chromosome there is a very strong locus, which or maybe close by loci that actually affect very much the resistance to this disease. You see that 40% of the genetic variant of the component, the genetic component of resistance, is explained by just this single region in the genome in linkage group 12. So this is certainly for us interesting. And so we moved to a second, in parallel, a second experiment, same F1, same parents, but in this case, we infected the fish in the same way, but instead of waiting for them to die, we stopped the experiment at 48 hours when the fish were still all alive. And then we sacrificed them and dissected the brain, which is a target a tissue organ for the disease, and the head kidney, which is the main immune organ for fish. And then we did a full-scale RNA analysis, RNA-seq analysis for all controls. 100 fish were mock infected with PBS, and another 200 fish were infected with in the same way than in the previous experiment. And here we found there was a correlation, was actually a association between genetic markers, again in the same region, for the expression of at least 2, possibly 3, depending on the tissue and the condition, controls or infected, in the same region of the GWAS for resistance, for survival. You see that we have signal also without the infection, just in controls, in the same region. So clearly, there are some variants, some EQTL controlling the expression of at least 1, as I say, possibly 2, maybe 3 genes. The problem when we went to look into this region, which was of course of great interest for us, we found a bit of a puzzling evidence, because what we were looking for was genes, coding regions, and possibly flanking regions of different genes correlating with the expression of those genes. Because the idea was essentially to look for, in this case, regulatory sequences, because apparently the phenotype of resistance is controlled by the expression of a gene rather than the variants in the coding region. This is quite common. I think it has been estimated that in human genomics that 90% of the phenotypes, disease phenotypes, are actually caused by locating regulatory sequences, and they affect the expression of a gene rather than the coding region. So the idea is, yes, probably the real mutation or mutations causing the resistance is in this red part, and we have to find a way to identify those red parts and to take away the linked SNPs, which are significant but not relevant for the phenotype. And as I was saying, when we zoom in into this region, we had a puzzling evidence, because looking at the first, of course, in the ensemble, and we found this quite complex annotation, where we had very long, for instance, here is a very long 5' and untranslated regions. And you see our smaller genes here. And there was something odd in the way the RNA-seq reads were mapping on these data. So we look at this is the very same sequence, genome sequence, just a different annotation pipeline in NCBI. And you see here, it is a bit more simple, actually simpler. You see this gene, it is this, but there is no very long 5' part untranslated. There is an additional one, which possibly is masked here by the annotation in ensemble, but there is a missing gene here, here, which is present here. And so we were a bit, confused, and we tried to look at independent evidence, and we actually ended up using data that have been produced in another work package. This was the work I'm presenting is work package 5 in AQUA-FAANG, and we used data from the work package 1, the body maps, and from work package 3, the immune maps that Oscar has already quite well described for turbot. The same thing has been done for all the 6 species, including the Seabass. So we ended up having, at least for this region, like what I call a Frankenstein annotation, merging the NCBI plus the missing gene that was present in ensemble. And in fact, this Frankenstein annotation for this important region for us seems to be supported by data from actually both ChIP-seq and ATAC-seq data. These are, I think you're familiar with from the presentation of Oscar. These are HMM data. These are from the head kidney stimulated in vivo with poly-IC, and this is the brain. And these are actually the RNA-seq data of the very same samples. You clearly see that this gene, which is missing in CBI, is a true gene. We have an active promoter here, at least in the immune-stimulated head kidney, and we have some evidence of some activity also in the brain. The RNA-seq data confirms this, and this gene and possibly this is also very active in the brain. So we decided this actually was a potentially correct annotation, and we moved on this, essentially trying again to find the red spots in the in our candidate region for the control of expression of these genes, and therefore the phenotype of resistance. And we thought that actually, based on literature, independently, irrespectively whether you are an enhancer or a promoter, or whether the promoter is active or not, normally you have open chromatin when genes are either active or ready to be expressed in a sort of permissive chromatin. So we focus, rather than looking at the ChIP-seq data, we decided it was better to focus on simply accessible chromatin regions. And in the same transcriptome RNA-seq expression experiment, also we in parallel, the third component of the experiment that was this complex experiment was to take out 10 fish from the control group and 10 fish from the infected group. And for those fish where we also have already RNA-seq data, also to have ATAC-seq data. And then we use those data to find open chromatin regions in the QTL area of fraction of the genome. And this is the result. We reasoned that comparing the two tissues would be interesting. In fact, we have 3 open chromatin areas in this candidate region. You see, these genes are actually quite interesting because they are copies of the same ancestral gene. We have look at different other species. We have 1 copy in some fish species. We have 2 copies in the sea bream, for instance. We have 4 copies in the closest relative of present which has a genome sequence is the striped seabass, has 4 copies. Actually, the seabass, the European seabass, seems to have 6 copies of this interferon responsive gene 27. But we had at the chromatin level, we had an open region just in the brain, and 2 which are actually in common between the brain and the head kidney. And there were very little differences, as Oscar already pointed out, for the ATAC regions, ATAC-seq peaks, depending on the condition. So the infected and the controls have almost the same peaks, actually the very same peaks. The only difference, and not as big difference, is in the between tissues. So we crossed this information of accessibility, having these as candidate regulatory regions in the genome based on the ATAC-seq peaks. And then we had 31 significant SNPs based on the GWAS for resistance, day to death, in the same region. And this actually led us to sort of pinpoint to single out 3 potential causative SNPs, 1 in this region and 2 in this one. The next step, which is still to be done, is to try to see if these three variants, has an effect on transcription, factor binding, but this is not easy because there are many softwares available, but it's not. Most of are based on human data, ChIP-seq data. So it's not an easy, and for vertebrate, I think the evidence is mostly from model species, so there's very little information on this. But ideally, once we are able to cross all this information, to integrate all of them, we might be even more confident that we have identified the regulatory causatives SNPs for this important QTL, for this important region, contributing 40% of the resistance to this disease. And that's all I have to say for my part.
Okay. Well, thank you very much, Luca. Any questions from anyone? I guess I have a question, which is in terms of the different data that you used, which did you find the most important for identifying the regulatory regions?
Well, as I say, I think for the purpose of genetics or identifying the causative variants, I think that the ATAC-seq data are probably the most promising, because I think they're more consistent across tissues and conditions, so you don't probably have to look at so many different cases or situations. And on the other side, those are, based on literature, really the best candidate for regulatory sequences in the genome. So I would focus on those. Maybe we have actually those data for both work package 1, the body maps, the dev maps. So now the next step is to broaden the approach and to use the same partition inside or outside the ATAC-seq peaks to sort of prioritize variants, not just in one region, but in the entire genome. So that's the idea. What it was a bit wanted to ask you, actually, as well, maybe it's not you're in charge, but I was a bit puzzled by the big differences in the annotation of the two pipelines, and honestly, it's a bit worrying to me, because of course we can sort of manually curate one region. We cannot really do that for the entire genome unless we start to do probably what Oscar was mentioning at the end of his presentation. So using ChIP-seq and RNA-seq and ATAC-seq data to refine and improve the annotation, which is, I understand it's an automated pipeline. You probably have, not you, you as EMBO, optimized to work on many species. So so do you have any suggestion, or is there any plan to revise the annotation based on?
Are you talking about the annotation you showed in your presentation in terms of the transcripts? You're talking about the gene annotation, or the?
Well, gene annotation certainly would help, because I mean, you see, if we do a correlation between gene expression and variants, if you're mapping your reads, your RNA-seq reads against something which does not exist, it's a bit useless.
Yeah,
You know
so
what I mean?
in terms of the gene build pipeline, so we should have recorded talk yesterday. And as you say, it's standardized across different species, but I believe they were using the data coming out of the AQUA-FAANG, and I think that might have improved some of the annotation, but I can ask Leanne some more about that and your case in particular.
Well, we found actually when we had the first ATAC-seq data for the entire genome, comparing, there is another version also in Ensemble, of the Seabass genome. And those was annotated, I think, 5 or more years ago. And actually, we found that the ATAC-seq identified the transcription start site were very good in the old genome, but in the new annotation, in the new genome, the actual predicted transcription start sites were actually upstream than what the ATAC-seq. So maybe there was a problem in the prediction of the 5' UTR. That's my very naive and very, let's say, preliminary interpretation. But that, because clearly I had the impression that either the data were the same, I guess, even less in the first annotation. So maybe the pipeline was slightly changed or significantly changed. So that's something that, I mean, I mentioned this in one chat with Peter, but clearly I understand this is just, you know, almost fully automated process. So we have maybe to clear, you know.
Yeah, but I think at the same time, you know, the edge cases, they can help improve the pipeline for all species. So I think it would be worth looking at this specific case. So I'll talk to Leanne and see what she says.
Okay, great.
Yeah.
Thank you.
Sure. Thank you. Okay, so I think maybe we could have a 5-minute break and then come back for ensemble biomarks. Elena, does that work for you?
Yep, that works perfectly. Thank you.
Okay. Thanks again, Luca and Oscar. Thanks for the talks. Bye then.